Our project focuses on predicting RNA binding protein binding from sequence using attention models. First, let me introduce what is RBP. RBP are proteins that bind to double or single-strand RNA in cells. Here is an example of RBP. Each basic domain recognises RNA, but the domain of most of these proteins require multiple copies to compare with their functions. RBPs especially play a critical role in post-transcriptional control of RNAs, so it is very important to predict them. ECLIF is an experiment which maps the binding sites of RBPs. Following this protocol, the PALD and CD fragments are amplified and sequenced. These peak regions are RBP binding sites, and the control groups do not have any RBP. Filtering by ECLIF experiment in ENCODE dataset, we find this HepG2 cell line. They are cells isolated from a hepatocellular carcinoma of a 15-year-old white male youth with liver cancer. There are 83 RBPs in total. We chose first 10 and then download the dataset using reference genome GRCH38 and file type and an arrow pick. Then filter out the 60,000 positive and negative region sorted by enrichment score to reduce computation. Then we'll split them into training and test dataset. This is a distribution of RBP sequence length. This peak region is manually set as 101 because it can cover most of these RBP binding site sequences. This is achieved by finding the center point and go up or down 15 nucleotides. After this preprocessing, the sequences are converted into one hot encoding matrix, where the I is the index of nucleotides, J is the index corresponding to ACGU in matrix. For the padded nucleotide at the start and end of sequences, we use 0.25 and use 1 if it's AGCU, use 0 if not.
So we use conventional CNN, CNN plus LSTM, and ResNet9 as benchmarks and utilized a 1D multi-attentional CNN with spatial temporal self-attention to explore how attention mechanisms help the task. We use 2D sequences instead of 3D data stitched by several repetitions of local sampling due to the short length of the selected RN binding protein sequences. The 1D multi-attentional CNN is first proposed in Wang's work to learn discriminate features in well-set bearing fault diagnosis. Two main attention modules are introduced. First is the channel attention module. Following the fusion of the nucleotide alignment information by the CNN, distinct channels may be employed to generate filters that match different motifs. The attention is calculated by 3 steps. First, performing global average polling on temporal dimension, and then pass through a CNN that cuts the number of channels in half. Finally, another CNN with sigmoid activation is used to convert to the original channel and obtain the attention weights. The recalibration is multiplied by original data, but to enhance the model's response to deep features, a residual connection is added. So with CAM, network sensitivity to diverse features as well as its interdependence among channels are improved, allowing it to adaptively discern the importance of features. The second module is the excitation attention module. Unlike the CAM, the EAM is intended to learn the nucleotide sequence associated with certain patterns. It can not only increase the efficiency and reliability of feature learning, but also make the network's feature learning process more understandable. First, use a convolution with 1x1 filter and sigmoid activation on input X to project features to temporal dimensions, and then to avoid over-focusing on excitation impulse signal segments. The feature information from X is encoded using a CNN prior to calibration. Also, a residue connection is added. So, as for an overall architecture, 3 successive CNN, AM, and CAM, and the dropout is referred to as one joint attention module. Within a JAM, temporal and spatial self-attention are calculated independently. The entire module architecture uses 5 JAMs to extract features, which are then globally average pooled before regression, the classification information. The module's configuration is also as shown, so we expect that the multi-attention 1D CNN can effectively integrate long and short-term information between sequences and nucleotide types and make training simple and convenient.
Now let's look at our results. This table shows the AUC comparison among these 4 methods on 10 different RBPs. The bold face indicates this performance is the best among the compared models, and we can say on the most experiments, multi-attention 1D CNN has the best performance and yields the best average AUC of 0.773. Then we employ multi-attention 1D CNN to identify RBP binding motifs across all datasets. For specific RBP, HNR-NPL, the left figure is a predicted motif, and the right figure is a known motif. It is obvious that the predicted motif for HNR-NPL is compatible with the well-known AC-rich motif, and the E-value is 0.00078. The results are quite good. In our study, we implement 4 different methods to predict RBP binding sites from RNA sequences. The multi-attention 1D CNN has the best performance overall. However, there are a lot of things we can do for future work. We can use more RBPs to train and test our models. Additionally, note that the positive and negative sequences are manually set to 101 nucleotides. However, some negative zones will eventually be defined as positive, and some positive zones may be defined as negative. So we can change the positive and negative region lengths and see what happens to the results. Finally, in our study, the prediction of RBP only accounts for nucleotide sequences. However, RBP RNA interactions also heavily depend on the structure and conformation of RNAs. So we can further predict RBP binding using not only sequence, but also structural information. This is all of our presentation. Thank you for your listening.